Cell organelles. The cell organelles are nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. An organelle is a specialized subunit within a cell that has a specific function and is usually enclosed within its own lipoprotein membrane. Except nucleus and chloroplast, all the organelles are seen only with the help of electron microscope. Various cell organelles are embedded in the cytosol. They are organs of a cell. Each organelle is living and performs a specific function. It has a definite shape, structure, and is bounded by a membrane. The membrane keeps its contents separate from the surrounding cytosol. More evolved cells and cells of multicellular organisms have to carry out great deal of metabolic activities. Membrane of each organelle helps to keep these activities separate. Most of these organelles are common in plant and animal cells and carry out the same function. Students, let us see each of them one by one. Nucleus. Students, do you remember the dark spherical bodies seen under the microscope when you observed the cells of the onion peel stained with iodine? Those structures are called nuclei. The singular form of nuclei is called nucleus. Let us now observe the nucleus of an animal cell. Nucleus is a large spherical body generally lying at the center of a cell. It is the most important part of the cell. It controls all the cellular activities and also plays an important role in cell division. It decides what function a cell will carry out after maturation. When observed with the help of an electron microscope, we can see nucleus covered by a double-layered nuclear membrane. The nuclear membrane has pores which allow the transfer of products from inside the nucleus or nucleoplasm to the outside or cytoplasm. The nucleoplasm contains one or more round-shaped nucleoli or singular nucleolus and a network of fibers called chromatin fibers. Nucleolus may be one or more in number and is not bound by a membrane. Chromatin fibers are thin thread-like structures composed of DNA or deoxyribonucleic acid and protein. These fibers condense to form short, thick fibers during cell division and become visible. DNA molecules transfer the hereditary information from one generation to the next. Functional segments of DNA are called genes. Generally, one gene controls one or more cell function, but sometimes a single function is controlled by a set of genes. Functions of the nucleus. 1. It controls all metabolic activities of the cell. If it is removed, the cell ultimately dies. 2. Nucleus controls cell division. 3. It is involved in transmission of hereditary characters from parents to offspring. In the red blood corpuscles of humans and other mammals, nuclei disintegrate, due to which there is more space to carry hemoglobin and to absorb more oxygen. In plant cells, like sieve tubes, the same thing happens and they become hollow for easier flow of materials. Endoplasmic Reticulum, ER. Endoplasmic reticulum is the intracellular transport system of a cell. Endoplasmic reticulum is a large network of membrane-bound, fluid-filled interconnecting tubules and sheets. Endoplasmic reticulum spreads from the nuclear membrane to the plasma membrane. Endoplasmic reticulum is of two types: rough endoplasmic reticulum or RER. It looks rough under an electron microscope as it has particles called ribosomes attached to it. Ribosomes. Ribosomes are present in all active cells. They are the sites where proteins are synthesized. Rough endoplasmic reticulum helps to transport the proteins synthesized by ribosome to that part of the cell where they are needed. Smooth endoplasmic reticulum or SER. Smooth endoplasmic reticulum is without ribosome and is meant for manufacture of fat molecules or lipids. Functions of endoplasmic reticulum. 1. Along with intercellular transport, it also acts as a supportive framework for a cell. 2. It synthesizes proteins and lipids or fats and transports them. Some lipids synthesized by it are used in building plasma membrane, while some other lipids and proteins are used as hormones and enzymes. 3. It transports proteins synthesized by ribosomes. 4. Smooth endoplasmic reticulum of certain cells of liver play a crucial role in detoxifying many poisons and drugs which enter our body through food, air, or water. It makes the toxins water-soluble so that they may be excreted through urine. Golgi apparatus or Golgi complex. Golgi apparatus was first described by a German scientist, Camillo Golgi. Camillo Golgi was born in 1843. He studied medicine in the University of Pavia. He developed a staining technique known as black reaction. With this technique, he was able to explore the nervous system in detail. In 1906, he shared the Nobel Prize with Santiago Cagel for their work on the structure of the nervous system. The Golgi apparatus comprises of 5 to 8 or even more sacs which resemble deflated balloons placed approximately parallel to each other. These sacs are called cisternae and they are filled with different enzymes. It is considered as the packing and forwarding department of the chemical factory of a cell. The proteins built in the smooth endoplasmic reticulum and rough endoplasmic reticulum come out enclosed in vesicles and move through the cytoplasm. When they reach the Golgi apparatus, they fuse with the cis phase or formation phase of the Golgi membranes and empty their contents in the cisternae. As they pass from fold to fold, they are biochemically modified by the enzymes present in the cisternae and are packed again in the vesicles. These vesicles exit from the transphase or maturation phase. Functions of Golgi complex. 1. Golgi complex is the secretory organ of the cell. 2. It modifies, sorts, and packs materials synthesized in the cells such as enzymes, mucus secretions, proteins, lipids, pigments, etc., and dispatches them to various targets inside the cell like the plasma membrane, lysosomes, or outside the cell. 3. It produces vacuoles and secretory vesicles. 4. It plays the role in formation of the cell wall, plasma membrane, and lysosomes. Lysosomes. Due to various metabolic activities going on in the cell, organic waste is generated. Lysosomes digest the waste. Lysosomes are simple one-membrane bound sacs filled with digestive enzymes. These powerful enzymes are made by rough endoplasmic reticulum and are capable of digesting and breaking down all organic matter. It is the cell's waste disposal system. Lysosomes are absent in plant cells. Functions of lysosomes. As it is the digestive system of the cell, it protects the cell by destroying any foreign material like bacteria and viruses that invade the cell. They remove worn-out cellular organelles and remove all the organic debris. This process is called autolysis. So, they are called as demolition squads. When a cell gets old or is damaged, lysosomes burst and the enzymes digest their own cells. Therefore, lysosomes are also called as suicide bags. Mitochondria. Students, now let us learn about the powerhouse of the cell. The organelle is called mitochondrion, which produces the energy for the cell. The plural of mitochondrion is mitochondria. Under electron microscope, a mitochondrion is seen as a double membrane structure. The outer membrane is porous, while the inner membrane is deeply folded. These folds are called cristae. They create a larger surface area for efficient functioning. The interior cavity is filled with proteinaceous gel-like matrix which contains ribosomes, phosphate granules, and DNA. Therefore, they are capable of making some of their own proteins. Mitochondria oxidize carbohydrates and fats present in the cells with the help of enzymes. During this process, large amount of energy is released, which is used by the mitochondria to produce energy-rich compound ATP or adenosine triphosphate. There are numerous mitochondria in animal cells, whereas plant cells have fewer mitochondria. Functions of mitochondria. 1. Mitochondria produce the energy-rich compound ATP, which is the energy currency of the cell. 2. The body uses the energy stored in ATP to synthesize chemical compounds such as proteins, carbohydrates, lipids, etc., and to do mechanical work such as movement of muscles, conduction of nerve impulses, production of heat, etc. Plastids. You must have seen leaves, flowers, fruits of different colors. Let us learn about the organelle that gives color to them. Plastids are double membrane structures present only in plant cells. They are of two types. 1. Chromoplasts, which are colored plastids. 2. Leucoplasts, which are white or colorless plastids. Chloroplasts are a type of chromoplasts which are green in color due to the presence of a green-colored pigment called chlorophyll. Pigment carotene present in the chromoplasts give orange color and xanthophyll gives yellow color. Chloroplast can get converted to other type of chromoplasts. For example, raw green tomatoes are converted to red. Chloroplasts are important for photosynthesis in plants. These are energy factories of plant cells. Chlorophyll traps solar energy and converts it to chemical energy. This energy is stored into chemical bonds produced during synthesis of food, that is, starch. Each chloroplast shows two distinct regions. 1. Stroma. It is the colorless proteinaceous matrix having various photosynthetic enzymes, DNA, ribosomes, and starch granules. 2. Granum. It is stack of thylakoids, which are the discs containing chlorophyll and other photosynthetic pigments. The tubes joining grana are called intergranal lamellae. Due to the presence of DNA and ribosomes, plastids can self-replicate like mitochondria. Functions of plastids. Chloroplasts help to harness solar energy and convert it into chemical energy in the form of food. Chromoplasts give color to flowers and fruits. Leucoplasts are involved in the synthesis and storage of various kinds of food like starch, oils, and proteins. Vacuoles. Students, let us find out how the cells keep its contents in an orderly manner. Vacuoles are storage sacs for solid or liquid contents. They do not have any basic shape or size. The structure of the vacuole changes according to the need of the cell. There are few small-sized and temporary vacuoles in animal cells, while plant cells have one or more large vacuoles. Vacuole is bound by a single membrane, that is, it is one-layered. Functions of vacuoles. Vacuoles help to maintain osmotic pressure of the cell. They store metabolic byproducts and end products, example, glycogen, proteins, and water. They store waste products and food in animal cell, example, in amoeba it stores food, so it is called as food vacuole. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to them. Prokaryotic and eukaryotic cells. Students, we have seen some important organelles of a cell. Cells having such membrane-bound organelles are called eukaryotic cells. In Greek, eu means true and karyon means nut or kernel, referring to nucleus. Characteristics of an eukaryotic cell. They have a well-defined nucleus, that is, with nuclear membrane, nucleolus, and nucleoplasm. Size of the eukaryotic cells ranges from 5 to 100 micrometers, that is, they are comparatively large in size. They have more than one chromosome. They are present in higher advanced unicellular and multicellular plants and animals. Prokaryotic cell. In Greek, pro means before and karyon means nut or kernel. Can there be cells without membrane-bound organelles? Cells present in prokaryotic organisms do not have membrane-bound organelles. Such cells are called as the prokaryotic cells. Characteristics of prokaryotic cells. Prokaryotic cells have three basic structures: plasma membrane, cytoplasm, and nuclear material. Their nucleus lacks nuclear membrane. Therefore, their genetic material, DNA, lies in direct contact with the cytoplasm. They contain a single chromosome. Such undefined region of the cytoplasm containing DNA is called nucleoid. Membrane-bound organelles, such as mitochondria, are absent in prokaryotic cells. In photosynthetic bacteria or blue-green algae, chlorophyll is present in vesicles and not in plastids. Prokaryotic cells are smaller in size, ranging from 1 to 10 micrometers.